So, welcome to the Synthetic Biology channel. We are iGEM-UANL 2012, and we've been here since morning, waiting for kids to come to visit our Synthetic Biology channel, which is basically an expo that explains what synthetic biology, biotechnology, and genetic engineering is. As you can see, children are coming all over the city to see what this expo is. The expo consists of 10 modules that cover topics regarding from DNA to ethic concerns about genetic engineering. The first one, if you want to go, this small demonstration of DNA extraction. It's basically the same protocol you find all over the internet to extract the DNA out of a banana. But yeah, we want to show it to the kids so they know how to do it and how easy it actually is. And then afterwards they move to the first module, which is DNA. You see a model of how DNA should resemble, and then we have our guest here who will explain to the kids what is DNA. We have our poster and also an interactive activity. But well, not all the children are meant to be here and to listen to what we have to say. The general public is also invited, and we prepare everything so that they can understand from the most basic concepts to the most complicated ones. So after DNA, we move to the gene module. Okay, our host is already exposing something.
Esto quiere decir que un gen se puede abrir la hebra por aquí y se copia.
To the interactive activity. Kids are supposed to build their own genetic construction here, which is composed by promoter, RBS, coding, and then the terminator, so that the children can know that DNA stores this information in a regular manner. So after they know what genes are, they go to the next module, which is genetic code in proteins. We got here some models of protein models, physically. And then our host will explain what proteins are, how they come out of a gene through the genetic code, which is there, this big circle, and then also an interactive activity, which is basically translating RNA to a protein. So after they know that genes are coding for proteins, they go to the genetic engineering module, where they learn that proteins can be used and also genes can be modified to allow a bacteria. They have there some models of to allow animal cells, bacteria, and plant cells to do whatever we want. Well, not whatever, but it's in the DNA that we are modifying and cutting and pasting all over the place. So in the genetic engineering module, they learn the basics of restriction enzymes, insertion, ligation, and cloning. So from all the concepts they already learned about DNA, about genes, and about how proteins are made, now they know how we can use them. So now we move to the fifth module. We're going to learn what synthetic biology is. We move now to the concept of DNA synthesis and standardization of genetic parts. Here is our host, Porfirio. Hi. and Hill. So that we already saw how DNA is stores information, how it's structured in genes, how it's then translated to proteins, how we can use genes and as a consequence proteins to build our own device or design devices, and how we can standardize these devices. So we move to the sixth module, which explains a little bit more what standardization of genetic parts is. We explain what biobricks are. Professor is already talking about what biobricks are in an interactive manner. We don't want to interrupt a lot here. So after they learn what biobricks are, then they move to the seventh module, where they learn what a biological machine is. How they can build something that works out of standardized parts like biobricks. So there's also Gilberto, our guide here. He's already explaining the interactive activity, what a biological machine is. Then we move to the eighth. After they learn that we can use DNA and genes in a standardized manner, and that we can build biological machines, they are meant to learn how can we apply all this that we learned. And so we come to the application section, where our guys here. There's Brennan.
Okay, we are in the application of biology, synthetic biology. This is in the medicine, is because the bacteria are in the body, but with the disease, we can have a way to identify the virus, and it's in the virus. But in the environment, we have some difficulties with these applications, and we can find the problems. Here is most application of this. Also, we have the industry, and it's in the genes, it's the color blue, and we have a way to identify all this color in vectors. Here is a chemical mode of found this. With the plants, we have other way to identify the places.
Yeah, you can be, yeah. Okay. So, we saw how can we apply a synthetic biological machine. We can apply it in all the different aspects that Brent already described: industry, agriculture, medicine, and environment. So after they learn where can they apply all this knowledge, we move to explain what iGEM is and what we've done. In our unit, we have been there in the iGEM for 2 years now. Last year we presented a project that won a silver medal. So, but yeah, it was not that much of more experimental work, but we did our best. Yeah, but the most important thing is that we present our work to a high school, so that they decided to form their own team for the high school division. And they participated in this year edition of the iGEM High School edition. And here's Lily from this high school called CDEP. That you'll see how many things they won. Lily?
Well, I am part of the CDEP team, and our project is about biosensor arabinose, it works in the whole way. If we have a high concentration, the bacteria will appear with a yellow color. If there is a low concentration, the bacteria will appear with a red color. And if there is no concentration, the bacteria will have a green color, which we call standby state. And this project had these results. We won the third place in all the world, and we won the best mathematical model. We were in collaboration with the iGEM-UANL team, and they have their project. Next, please.
All right. Well, from last year's project, what I believe is the biggest application that we made is this incubator, which was modified so that it can somehow, when we're culturing our plates, light will come up from this two sources, and here we see two mirrors that will reflect light
To the bacteria.
exactly to a place that we want in a plate, so that we can have a particular filter so that we can have light in a certain wavelength. And well, we were working with red and green light so that when we were exposing our cultures to green or red light, it will present a different behavior.
It has to enter two inputs and 5
And
outputs. Yes. And it's like an intelligent bacteria who responds to different light.
Exactly. And this year project is, we made out of the genetic circuits categories, and we made to the environmental one. And we're gonna present arsenic bioremediation device that consists basically on a bacteria that will suck the arsenic out of the solution, and then we will be able to extract all this bacteria that is loaded with arsenic with a novel silica binding device that is basically on a chimeric protein that will be built up out of OMPA and a domain that binds to silica that comes out of a ribosomal protein that is L2. And we hope that to do a very good presentation there in Bogota. So yeah, to all the friends in the Latin American edition, hello. And then we move to the last section. That is about, the ethics and biosecurity concerns of genetic engineering. We explained to kids after we explained that we can use genes to build up biological machines, and we can use them to a lot of applications. Well, we tell them, okay, not only because we can do something means that we should do that. There are so many concerns that we have to take into account. And then after they learn that, biosecurity has to be taken into account, environmental protection has to be taken into account as well, we give them survey so that we can see in a quantitative manner what they've learned from all this exposition that we are making. And then we move to the last place. We're moving to the last section of our channel, which is basically just a goodbye part where kids and all our visitors can write whatever they want. Well, we're supposed to have here a very large piece of paper, but okay, in principle here should be a large piece of paper where kids can write their names and just say, hey, we've been here on the Synthetic Biology channel. And of course our seminar story.
Hi.
And we hope you enjoyed the trip through the Synthetic Biology channel. We're expecting like 400 around 400 kids per day. They are already confirmed, and many schools from the from the public district here in our city, Monterrey, and yeah, and also from some private. Well, we call them secondary schools, which we basically junior high in the United States, and well, we're expecting around 400, as I already told, 400 kids, and also, well, people that are just wandering around and want to see, well, what's going on here? What is all these people doing with biology and stuff? So maybe, who knows, we'll be having around 600 people here, and our host will be presenting all this, all these models that we prepare actually by our own hands. Here is Areli, who has been Kelly helping me holding the laptop, and well, we hope to see you there in Bogota for the Latin American section, and hopefully also in Boston. So goodbye, we are team iGEM-UANL 2012, and well, it was nice being with you.